Hello and welcome to the veterinary anatomy brainry, and today we'll be discussing about the histology of nervous system. So if you look out the nervous system, the basic cells that are present in the nervous system will be of two types. One are the neurons and one that is the main cell or the principal cell of the nervous system. And the supporting cells will be there, and collectively you call them as neuroglia. And the basic structure of any kind of a neuron is that it has a body or called as a soma or perikaryon. Then it has the single process that is going from the body you call as an axon, and then there are the multiple processes that will arise, small processes that will arise from the soma. You call them as dendrites. The features of perikaryon or the soma or the cell body is that it attains the central position in case of the neuron, and the shape basically is the polygonal, and the size and the shape of this cell body that is determined where and to which part these neurons will belong. It will have the different shapes and the size. It will contain the cell organelles. It will have the nucleus, then it will have the cytoplasm and other things like other cell. Then the process, what I told you, is the main is the axon, and it's a single process that will be arising from the soma. Then the dendrite, small process that will be arising from the soma. You call them as the primary dendrites, and from primary dendrites you have the small spiny like structures. You call them as spiny dendrites. And if you look out at this staining slide, you can see here this is a soma with that of nucleus and nucleolus. Then the axon is arising. You call that area as an axon hillock, and you can see the axon is also there. And this is the connective tissue, small part of connective tissue that is present here, and these will be some of the supporting cells also. If you go for the classification of the neurons, you have two, three types of the two, three classifications. One of the classification that depends upon what is the size and the shape of the processes. You have the multipolar neurons. So many processes will be there in this type of neurons, and these type of neurons usually are motor in nature. Then you have the bipolar neurons, and basically these are in present in the retina. You have the two processes that will be arising from the soma. Then you will have the pseudo unipolar. It looks like that of a unipolar, but it is not, and it looks like that of a T. So it's called as a pseudo unipolar neuron. You have the sensory ganglions as an example. And one more classification depends upon whether a neuron is the sensory or motor, or whether the neuron helps in the interconnection between the motor and the sensory. So sensory neurons, also known as the afferent neurons, where the impulse will be received, it will be conducted towards the CNS, and action will happen. Then you have the motor neurons, where the CNS generates this information or the impulse that will be going towards the muscle gland and other neurons for the functioning. Then, if you look out the neuronal groupings, the presence of the neurons in the different parts of the CNS, if you talk about the cerebral cortex, or if you talk about the cerebrum and the cerebellum, the neurons are basically arranged in a particular manner in the layered fashion, and that's why in case of cerebral cortex, you have the six layers of the six layers of the neurons. In case of the cerebellum, you have three layers of neurons, while if you look out then other regions as the subcortical regions, as the thalamus, midbrain, brainstem, you will have the neurons as in an irregular cluster-like fashion. So that means there will be no layering of the neurons in the thalamus, midbrain, and other parts of the brain. Then ganglions, those are basically the cell collection of the cell bodies outside the CNS. Again, they will be in the form of the clusters, not in particular manner like that layering in case of cortex. The supporting cells, they will are generally more than that of the neurons, considered as 10 times. In case of humans, they are 10 times more than that of the neurons in the brain. And these type of cells will go under the mitosis, and they will be responsible for any disease condition of the brain. These are the main cells that, because they undergo the mitosis, while the neurons, they lead the form this the mitotic feature. And the main function of these neuronal cells is that they will just provide the structural support to that of nerve cells. And if you go for the classification, you will have the cells that are present in the CNS. You have the oligodendrocytes, astrocytes, ependymal cells, microglia. Then in case of the PNS, you have a cell, a supporting cell as a Schwann cell. And if you see the astrocyte structure of the astrocyte, basically it has two types. One is called as the fiber astrocyte, one is called as protoplasmic astrocyte. The main thing is that in case of fiber astrocytes, the process will be very long. And the function of this astrocyte will be that it will be controlling the ionic and the chemical environments of the neurons. Then one more type of the cell will be the microglia cell that has a phagocytic nature, and basically that microglia is also known as Hortiga cell. That is the name of the scientist who has discovered it, and they are basically derived from the mesoderm. And if you look out, one more cell will be there present which produce the myelin, that is the oligodendrocytes, and this is the cell that produce the myelin sheet around the neuron, and also and the nodes of Ranvier will be formed by the oligodendrocyte cells. Then ependymal cells, the cells that will be lining the ventricles of the brain, and even they will be lining the spinal canal. Those are the ependymal cells. They help in the formation of the CSF. Then the outside are basically the PNS, peripheral nervous system. You have the type of supporting cell or neuroglia cell. You call it Schwann cell. It is similar to that of oligodendrocytes. Here it produces the myelin for the nerve fibers. A basic classification of the nervous system is you have CNS and the PNS. CNS itself is divided into cerebrum, cerebellum, spinal cord, and there will be a less or no amount of the connective tissue that will be present in the brain. That's why it looks like that of a jelly-like thing. And when you cut the brain, you will have the two parts, white matter and the gray matter. And this white matter and the gray matter is basically of the staining procedure. If you stain the brain, a part of the brain will not take the stain, basically the white matter, because this will be made of the myelinated neurofibers, and the myelin will not take the stain, and it looks like they're white. And the gray matter basically takes the stain, mainly made up of the soma, the cell bodies, and it will take the stain, and on that basis so you have the name as a white matter and the gray matter. Even though on the gross observations, if you cut the brain, we will look the two colors or the two parts of the brain clearly as a gray matter. On the picture you can see outside is the gray matter and inside is the white matter. All these structures of the nervous system will be covered by the meninges. Those are the very toughest coverings of the brain, besides the skull, besides the bones that will be covering the brain. So after these bones, there will be other coverings. You call them as a meninges. So looking out the histology of the cerebrum, I told you that two things you can find under the microscope also, gray matter and the white matter. And the gray matter, usually you have the neuronal cells, small dendrites and the glial cells. And this gray matter basically is half, I told you the cells will, neurons are arranged into six layers. Then the white matter basic is that the myelinated axons will be formed. So before going to the six layers, you should know that in the cerebral cortex, in those six layers, there will be the different types of the cells present. You have the neuronal cells in the cerebral cortex as pyramidal cells. And you can look out here, the pyramidal cells here. This is a pyramidal cell. And they are more superficial. So more superficially, so in the layer, if you see out, it is present in the layer third, basically. More are in the superficial, some will be in the deep region, like in they will be more present on the superficially. And if you have same type of pyramidal cell, large, that's called that cell, so they will be just in a deeper position. So large cells, so it has two, one is small cell will be more superficial, large pyramidal will be more deeper in. Then one more cell is satellite cell, also known as granule cells. They are the star shaped cells. So this is a star shaped satellite cell. So they are basically connected to the near neurons. Then you have the cells of Martinotti and axons to the superficial layers. You have the here. So the axon of this cell will be in the superficial. You see here the axon will go towards the layer first here. You can see here, even though the cell body is present in the layer third. Then you have the fusiform cells, spindle shaped. You look out here, the shape of here, the soma, it is a spindle shaped cells, and the orientation of these cells are the vertical orientation, and this type of the cell will be in the deeper position. Then you have the horizontal cells of Kajal, and again you have horizontal cells here, again the spindle shaped cells, but here these cells will be in the superficial layers. So both the cells will be this fusiform, and horizontal cells of Kajal both are spindle shaped, but horizontal cell of Kajal will be in the superficial layer as layer first you can see, while in case of the fusiform it will be in the deeper position. So accordingly you can name the layers, like layer first will be having the horizontal cells plus the axons of the Martinotti cells. Layer 2 will be mostly having the satellite cells. Layer 3 will be having satellite cells plus some small pyramidal cells. Layer 4 will be having the satellite cells and the axons of the other cells. Layer 5th will be the fusiform pyramidal, and layer 6 will be morely of the fusiform cells. So if you look out the histological picture under the 4x, you can see like this, and you can name the layers as 1, 2, 3, 4, 5 as molecular layer, outer granular layer, pyramidal layer, inner granular layer, ganglionic layer, and the multiform layer. So these are the 6 layers that are present in the cerebral cortex. That means that these neurons are arranged accordingly these 6 layers. So in this also you can see that the representations of these layers, 6 layers. The multiform is also known as paleomorphic layer here also. So this is again the H&E stain 4x. So this is a different type of stain, as you can see the how the neurons, the neural layering is there. So these are the description of the 6 layers, and what I told you, the first layer you will have the horizontal cells more. The second layer you have the this satellite cells again here. No, sorry, in case of fourth, in case of the second, you have the pyramidal cells. In case of third, you have 2 layers of pyramidal cells. So accordingly, different types of cells will be present in the different layers. And if you look out the 6th layer, you have the spindle shaped cells, Martinotti cells, satellite cells. Almost all of the cell will be present in case of the 6 layers in the 6th layer. Then that was the histology of the cerebrum, where the neurons are arranged into the 6 layers. In case of cerebellum, basically you cannot find the lobulation of this hair also, nor the division. But under the gross section, you can find 2 things where the white matter will be arranged in time of the, like you can see here, the white matter goes like that of a tree. Call them as a folia arrangement of the gray matter and the white matter. You can see here. So cerebral cortex, also known as the folia, you have the central nuclei as a gray matter. So tree of life or Boroviti, that is white matter will be arranged because here will be the cerebellar peduncle. Cerebral peduncle is basically the white matter that will go inside towards the parenchyma or the gray matter of the cerebellum, and it will divide like that of a tree. Tree of soul is also known as here. Here you can see the H&E stain, cerebral cortex, and the white matter, how it is arranged. Again, here in the cerebral cortex, I told you that the neurons will be arranged in 3 layers: outer molecular, central layer, and the inner granular layer. So this thing I told you has 3 layers. Outer molecular layer, you can see the cells are there are few neurons, and mostly these neurons will be unmyelinated. And there will be a cell line that's called central layer of the large Purkinje cells. So this is the enlarged view of Purkinje cell. You can see here Purkinje cell layer. So this will be the large cell layer as a Purkinje cell layer. Then there will be the inner granule layer. So these are the 3 layers of the cerebral cortex, how they are cerebellar cortex as outer molecular, middle Purkinje, and the inner one is the granule layer. Then looking out the spinal cord histology, where it is a different story than that of cerebrum and the cerebellar cortex, where the gray matter is inside and the white is outside. So this gray matter inside in the cerebral, in the spinal cord will take the shape of H. You can see here it is the shape of H here, and with the central canal at the center. And arrangement of these gray matter, you can see you call them as a horns. You have the posterior horn, you have the anterior horn. Then accordingly, the white matter that is surrounding here, here, and here, it is also the names. You have the different names as in the white matter also. So you have the anterior horns and the posterior horns. You have the central canal. So that is one of the difference between the cerebral, and the spinal cord, where the gray matter is inside and the white is outside. So again here, if you look out the anterior horns, usually they are the motor neurons, or they are the motor neurons basically. And Then there is a one term that we call as neuropil, because this is the meshwork of dendrites, axon terminals, and the process of the neuroglia cells you can find out in the white matter. That's called as neuropil. Now, if you look out the white matter, what I told you, like you have the name of gray matter as the horn. The white matter you can name it as a funiculus. So this side it is the posterior horns, so it will be the posterior funiculus. This side it will be the lateral funiculus, or here it is the name lateral funiculus. Same will be here lateral funiculus. And these are the anterior horns, so this will be the what you call as anterior funiculus. So anterior funiculus, lateral funiculus, and the posterior funiculus. Name of the, or you can say the division of the white matter in case of the spinal cord. So neuroglia cells that you find mostly in case of the spinal cord, oligodendrocytes, microglia, and the fibrous histiocytes beside the blood vessels that will be supplying to this also. Then the outer covering that we have discussed, three coverings will be there besides the bones, dura mater, arachnoid, and the pia mater. The outermost is the dura, it is called as hard matter. Arachnoid is the web-like structure. Pia mater is called as the softer matter. Then there is a spaces, subdural space under the dura mater. There is a space that called a subdural space. Then under the arachnoid matter, that's called as subarachnoid spaces. So if you take the histological section of these meninges, you will have mostly the dura mater made up of the tough collagenous fibers. Arachnoid will be having the collagen fiber, but not unlike that of the dura mater. Then the pia mater basically is called the vascular meninge. It is mostly attached towards the brain. Basically, it's a very delicate layer and made up of the mesothelial layer only. Now, PNS, if you talk about the peripheral nervous system, it has neurofibers, axons outside the CNS and surrounded by the connective tissue. So, main component of this peripheral nervous system, you have the peripheral nerves, you have the ganglia, and then nerve endings. So, this is the then the coverings of any type of the nerve. You have the epineurium, you have the perineurium, and you have the endoneurium. So epi means outermost covering. This will be the collagen connective tissue. Then peri means dense connective tissue, and then endoneurium is loose connective tissue. So here you can see here the HND stained peripheral nerve. So this is the nerve and this is the connective tissue. You can see here these are the nerve bundles, or this is the nerve, and there will be so many these nerve fibers will be present in this. So ganglia, you know that outside collection of the cytons outside the CNS, you call them as ganglia, and the main function of the ganglia is they act as the relay station to transmit the impulses. You have two types of ganglia, sensory and the autonomic ganglia. Then autonomic nervous system, if you look out, two parts will be sympathetic and parasympathetic that will act in, one is norepinephrine type and one is acetylcholine type. And this is basically the histology of the nervous system. And hopefully I will go through this. And if you have any questions, you can ask. You can ask through any media. You can ask. You can just comment on the channel or comment on the video if you have any difficulty in this video. Thank you.